Hello boys, welcome back to another interesting topic on video tutorials, and this session is meant for class 12th biology students, and this is meant for 20th August 2020, and our memory helps us in tracing out what we have been discussing and reflecting upon. We were talking about a very interesting experiment called Hershey and Chase blenders experiment in our previous session. Now we will be discussing about another interesting experiment which proves that DNA splits, and thanks to Professor Watson and Crick. They were the scientists who proposed first time double helix structure, and they also said that in between purines and pyrimidines there is a weak bond called hydrogen bond. And I also said, if you recollect from your memory, sometimes our weakness becomes our strength. So because of the presence of that weak hydrogen bond that can be broken, DNA replication takes place, and because of DNA replication, varieties will be formed. So thanks to the weak hydrogen bonds. Now in this session we will be talking about that exactly, how DNA replication takes place, what are the intricacies involved in that. So first, criteria for genetic material. First, it should be able to generate its replica. Replication should take place and the replica should be formed. It should be chemically and structurally stable. Many times you studied in chemistry, the ultimate aim of any chemical compound or chemical molecule is stability, and it should provide slow changes. Mutation should not be there that required for evolution. It should be able to express itself in the form of Mendelian character. We said, you see, the law of separation, law of independent assortment. Remember, protein does not fulfill the criteria, hence it is not the genetic material. RNA, DNA fulfill that above criteria, hence they are genetic material. This is theoretical. How about the proof? We need proof. Being biology students, always we go for the proof. And we are thankful to so many scientists. They have proved beyond doubt that DNA is the genetic material. Now, how does the replication of DNA takes place? What is it, first of all? It is the process by which DNA produces the exact copies of the original DNA. So, it is Xerox copies. In eukaryotes, DNA is double-stranded. The two strands are complementary to each other because of their base sequence, purines and pyrimidines. And semi-conservative method of DNA replication takes place. It is the most common method of DNA replication. It takes place in the nucleus where the DNA is present. Replication takes place in the S phase. Remember, in cell cycle we talked about various phases before actually prophase starts. S is synthetic phase. Okay, and deoxyribulose nucleotides needed for the formation of new DNA strands. I said polymer. What is DNA? If someone asks, what is your answer? It's a polymer of nucleotides. Okay, so DNA strands are present in nucleoplasm. So at the time of replication, two strands of DNA separates, as you are seeing in the picture. Each strand acts as a template for the formation of new strand. It's a template. A new strand is constructed on each old strand by complementary base pairing. As I see, in the nitrogenous base pairing, adenine should be always paired with thymine and vice versa. Thymine pair with adenine. Similarly, guanine should pair with cytosine and vice versa. Cytosine should pair with guanine. This is complementary pairing, base pairing. Okay. Hence, two exactly identical double-stranded DNA molecules are formed. In each new DNA molecule, one strand is old, that is a parental strand, while the other one is newly formed. Hence, Watson and Crick described this method as semi-conservative replication. You are seeing new one and the old one. Semi-conservative nature of DNA was this proved thanks to two scientists, again great scientists, Messelson and Stahl, it's called. Of course, the spelling is STAHL, Messelson and Stahl experiment. And they proved that DNA replication takes place through semi-conservative method. So what is that they have done? In DNA, of course, nitrogen bases will be there, and in nitrogen bases, the normal nitrogen is N14. But what they have done, they used their intelligence and used an isotope of nitrogen, that's N15, so that it can be heavy in nature and it can be sedimented. This is what they used. So they have taken E. coli DNA because uniformly labeled with a heavy dose of nitrogen, 15 M, and then in E. coli grown in labeled medium, okay. And in generation 1 they could see that because of gravitational force, so it is only all N15 N15. And then next generation N15 labeled E. coli added to N14 medium. Now that N14 is normal one, and the cells replicate once in N14. In generation 1 what happens? You are getting two strands. One strand consisting of isotope nitrogen 15N, one normal 14N. And in second generation you can see cells replicate in second time in 14N, and when DNA is extracted and centrifuged, it is observed that one DNA is the N14 strands, another DNA is having two strands with the N14 and N15. And when third generation, when we observe, cells replicate third time in 14N, and it is observed that you got 14N, 14N, 15N, 15N. So we got all varieties here. Now they have taken even the bands photographs, and this is how the scans were observed. So starting it is 15 nitrogen containing medium, E. coli cultures of course. Basically all these experiments are being done on a bacterium, Escherichia coli, which will be present in our small intestine. So the DNA is graded with the help of a chemical called cesium chloride, CSCl. So first you can see the DNA composition. Heavy DNA is formed. Both these strands are 15N, 15N. And when the replication cycle starts and the culture is done in 14 nitrogen, the first generation you have got a blend, that is intermediate density DNA, 15N and 14N. And when you go for the replication cycle 2, in the same culture continuation, and in the presence of cesium chloride, the DNA is gradient, and we are getting two DNAs. One is lighter DNA, 14N, 14N, both the strands. Another one is intermediate density, one strand 14N, another strand 15N. And when replication cycle third starts, it is observed that varieties are formed. What is that? 14N, 14N, first one, 14N, 14N, because parental one is duplicated. And in this, 14N, 14N and 15N, 14N. So this is how we can prove that it is, the replication is semi-conservative. Like again, this is explained in this way. So basically it is isotope 15N DNA is taken for 20 minutes, that culture medium. So first generation you got 14N DNA, one strand, another strand 15N DNA. Another 40 minutes it is observed generation 2. So 14N DNA, lighter DNA, 15N DNA and 14N DNA. It's a hybrid DNA, and then normal DNA. So heavy DNA is formed first in a centrifugal machine. So it is recorded at the bottom, and hybrid one is formed in the middle, and the light one is on the supernatant, and the heavy or hybrid one in the middle. So separation of DNA is done by centrifugation method, and Messelson and Stahl proved beyond doubt that DNA replication is always semi-conservative. So parental DNA molecule you are saying having 15N and 15N. So first generation DNA molecule is got split up 15N and 14N because the culture that they have done is 14N medium. And then in second generation you are having a hybrid 15N, 14N and one lighter one 14N and 14N. So semi-conservative nature of DNA by Matthew, Messelson and Franklin Stahl. So what is that they have done? They grew E. coli on, of course, ammonium chloride, isotope nitrogen 15, culture medium. 15N is the heavy isotope of nitrogen. Both strands of DNA have 15N. That's why we said heavy DNA. These bacteria are shifted to ammonium chloride culture medium. DNA extracted subjected to cesium chloride, CSCl, and CSCl density gradient centrifugation is done. And what we got is hybrid or intermediate type DNA, 15N and 14N. After next generation, equal amount of light DNA, 14N, 14N and 15N, 14N are formed. So mechanism of DNA. The process of DNA replication takes place by number of substance, enzymes involved and proteins. They are Substances, deoxyribonucleotides. Enzyme, DNA helicase, DNA polymerase 1 to 3, RNA primase, and DNA ligase. And the protein is SSB, not Service Selection Board, of course. Single strand binding protein. Single strand binding protein. So this is how it happens. So you got template strands, and there is a replication fork, Y-shaped one, and the original DNA one, DNA polymerase